So my talk today is mostly centered on this paper that we published, I guess, about a month ago. If you're interested in reading the paper and if you don't have access to it, please just email me and I'm happy to send a copy out. It's no problem at all. The paper is called Positive Citation Bias and Overinterpreted Results Lead to Misinformation on Common Mycorrhizal Networks in Forests. So kind of a wordy title there. Basically, in sort of plain language, we ask this question of, the Wood Wide Web. What's happened to it? What's the story? Is it all true? And that's kind of where we begin. So, but before I start, I need to acknowledge my two co-authors, Melanie Jones, she's at University of British Columbia, and Jason Huxema, he's at the University of Mississippi. Melanie actually was my PhD advisor, and Jason, I did a postdoc with him, so I know them both quite well. And I honestly would not have been able to publish this paper without them. So they have contributed so much intellectually. And also, as you can imagine, there was you need to be working with people that you really trust. And these two, I trust with my career, and I hope that they would say the same thing. And that was really an important element of this particular team. So yeah, without those two, I would not be in the seat talking about this paper. Okay, so just so we're all on the same page, I want to, and I think everyone knows this, but I just want to make sure we're all talking about the same thing and what a common mycorrhizal network is. So a common mycorrhizal network is formed when roots of different plants, in this case, it's two different tree species, they're connected below ground through fungal tissue, and so it'd be mycorrhizal fungal tissue. And as Susan alluded to, we often call it the Wood Wide Web, and we can sort of talk a little bit about that later on in the talk. So I'm focused on looking at the structure and function of common mycorrhizal networks. And this Wood Wide Web, as you know, it's captured the interest of broad audience. Everyone seems to know something about the Wood Wide Web. And this idea that trees benefit from being connected through this below ground network, it's been popularized in books like this one, Finding the Mother Tree, The Hidden Life of Trees, The Over Story. It's been popularized in newspaper articles that you've probably come across, like this one that was in the New York Times, I think in 2020. It's also been showcased in documentaries like this one, Fantastic Fungi. The Wood Wide Web has been showcased in podcasts, TED Talks, and if you're listening carefully, you might have also heard it alluded to in Ted Lasso, if you're watching that series. But it doesn't just stop there. So for example, last Saturday, if you were doing the mini crossword in the New York Times, you might have come across this. The clue for number 6 down, organisms that help trees talk to each other underground. And then, so of course, fungi. It's not just in books for adults. There are several books out there for kids that are teaching them about how trees communicate below ground through fungi. I'm a little bit closer to my home, and maybe some of you on the Zoom visited this park. This is from the Malahat Skywalk in British Columbia, Canada. And this sign, so this is a park interpretive sign, and you could probably see, so it says, no Wi-Fi, no problem, wonders of the Wood Wide Web. And that little small text, I'll just read it out for you. It says, mother trees send nutrients to saplings to give them a boost. Dying trees shed nutrients to donate to others. A tree under attack sends a chemical signal to warn others to boost their defenses. So, and this is an interpretive sign in a park. And it doesn't just stop there. So in March alone, UN Biodiversity has puts out several tweets showcasing the Wood Wide Web. So showcasing the social networks of forests, the role of the root system in communicating with trees. So this is, it's everywhere, literally. And this is something new that I found. Someone actually sent it to me. So this is a newspaper article. It's out of Pittsburgh. And this was out on March 12th. And here, so most recently, the Wood Wide Web has been used to argue why forests shouldn't be cut down. So I'll just read a line from it here. It says, an environmental group is suing to stop the US Forest Service from moving forward with the project that would clear cut 1,600 acres in the Wayne National Forest. This project would destroy the Wood Wide Web, which communicates with trees underground. And here the attorney in this case is arguing that the importance of these networks is well-established science. So basically, you know, probably like many of you, we were hearing extraordinary claims about common mycorrhizal networks in forests, what they do. And it made us, these extraordinary claims, it just made us wonder, are they true? And I think for Jason, Melanie, and I, when we first started kind of hearing about these, we all had the same reaction of, we all felt that we must be behind in the literature. And you know, there's just so much coming out all the time when you're a scientist, and it's hard to stay on top of the literature. And so that's what we felt. We're like, there must be some groundbreaking studies that have come out that we just have not come across yet. And so our first instinct was to, okay, yeah, we need to check because we must be missing out on something. So in doing that, our purpose was actually pretty simple. We wanted to evaluate some of these popular claims. Do they hold up to scrutiny? Are they simply true? But also, we were interested in investigating how scientists interpret some of these claims about the Wood Wide Web. So there's going to be two parts to this talk, kind of focusing on each of those purposes. So first, we wanted to get a sense of how the public was thinking about the Wood Wide Web. How are they talking about it? How are they thinking about it? And so to do that, we went through a lot of the popular media. And right away, we discovered that there's just so much out there. And even after we published the paper, people were still sending us, oh, you should have looked here, you should have looked here. Like, so we did not capture it at all. But we did get, I think, sort of the bestsellers, what we thought were getting big audiences. So we read 10 books. 4 of these are bestsellers. We came across 4 kids' books. We listened to 11 different podcasts, watched 11 short videos. You've probably seen some of these videos. Some of them are like BBC, PBS, went through 25 magazines or newspaper articles, watched 6 different films or documentaries, and then sat through 2 TV series. So this alone, we added up, it was about 150, 200 hours of just going through the popular media. And again, it was to get a sense of like, how are people thinking about this? So from that, we found that there were 3 claims that were popping up over and over and over again. And so the first one is this idea that common mycorrhizal networks are widespread in forests. The second one is that resources are transferred through common mycorrhizal networks, resulting in increased seedling performance. And this idea that mature trees preferentially send resources or warning signals to their young, or sometimes it will be called kin, through common mycorrhizal networks. If I slip, I might say network, I might say CMN, just know that I'm talking about common mycorrhizal networks. So this first claim that common mycorrhizal networks are widespread in forest, I want to be really clear that we are not disputing the importance of mycorrhizas in forests. So there is lots and lots of evidence showing that mycorrhizal fungi are really important to tree nutrition. The establishments of seedlings, they're really important in soil formation. I don't think anybody disputes their importance in forests. So that's not what I'm talking about. Here, we were really focused on these connections, the importance of these connections between trees or trees and seedlings. So I just want to be really clear so no one goes away and says that Justine said that mycorrhizal fungi are important. That is not my message at all. So again, I'm really focused on these connections between trees. And you probably kind of heard it talked about it in different ways. Like a really common metaphor is to talk about the internet of the forest. And so it paints this picture that all trees in a stand or in a forest are connected below ground, and the fungi are acting as these passive conduits, just sending messages where the trees need, kind of thing. And on one hand, you know, when you think about tree roots and when you think about just how much or how many mycorrhizal fungi are in the soil, we think it's very plausible and it's very likely that trees and seedlings are connected below ground. We don't really dispute that idea, but it's this idea that they're widespread in all forests is what we're focusing on. So, and then as many of you probably know, it's difficult to map out a common mycorrhizal network in a forest because as soon as you start digging in the soil, you of course destroy the network. So we came across one study, and some of you might know this person, Jim Trappy. It's an older study from the 70s, and it was the one case, and it was a really fun paper to read because he went out and he did, dig up roots and then find I can't remember what fungal species it was, but it was something that had more of a like robust mycelium. And then he actually followed it in the soil to see which roots it was connected to. It's really, really nice detective work, and it's really, really nice natural history, but no one else has done this. And so for the most of us, we have to use indirect methods. So we can't dig up the soil to map this mycorrhizal network and see which tree is connected to which tree below ground. We have to use indirect methods. And so what this looks like is what we call genotype mapping. And so I personally have not done this. So I'm going to talk about it. If I say we, I actually don't mean me. I'm going to talk about researchers. The way that this works is you can go into a stand, say like this Douglas fir stand, and most of the tree stems are all Douglas fir. So you don't have to worry about which species you're dealing with. It's all the same species. And you can take a sample from this tree, say from the bark or the leaves, the needles, and you can sequence the DNA so that you can get a unique marker for each of these stems. So I know which genotype each of these trees are, and I know that they're different genotypes. I can then dig in the soil and trace and find roots. And Douglas fir roots, when they are colonized by species of Rhizopogon, they form these little tubercules over here. So they look like little potatoes in the soil. So if I come across these tubercules, I know that they're specific to Douglas fir. So I know that these roots that they're attached to are Douglas fir. And I know this is Rhizopogon by this little structure. So it's kind of an interesting mycorrhizas because it's one of the few that forms a structure that you can actually see and you can like identify. So if I find these little potato looking things, I can then genotype this root so I know what stem it belongs to in the forest. And then I can also genotype the fungi so I know which individual that this fungi belongs to. And then I can keep track of the locations in the forest. And so I can build this map. And it's important to know which genotype of fungus we're working with because only the genotypes can fuse. So different species of fungi, they're not going to fuse and make a network. It's only identical genotypes that are going to do, or that can fuse like that. So from using these techniques, there's been some really beautiful maps developed. So this is one of them. This is by Byler et al. And it came out in 2010. And you're looking down, and this is a Douglas fir stand. So the green little dots, these are different Douglas fir trees. The size just tell you how big or small the tree is. These little black dots that you can see in the picture, this is locations where they sampled those ectomycorrhizal roots. And then the lines, these correspond to different Rhizopogon species and different genets that then are connected to the tree. So by doing this, it's basically you can kind of connect the dots. So what this research tells us is that some fungal genotypes colonize different trees, and that we know that a single fungal genotype can span up to 21 meters in this forest. So again, and I want to stress like how difficult it is making these maps. So I don't want to trivialize these maps at all or dismiss them. They're really, really valuable. But there is some limitations. So as I said, so we're taking discrete samples. And so what these discrete samples in space tell us, they show us a picture of the legacy of the common mycorrhizal network, but it doesn't mean that that mycorrhizal network looks like that right now. And so what I mean by this, I have a little picture. Okay, here's a Douglas fir tree. Here's another one over here. And here I've drawn on like pretend roots. And then this little pink blob. So this is a fungal genotype. So it starts growing, it colonizes these roots, and then it's going to expand. So then it grows out, and then it colonizes the root system of this other tree. So we would consider this a common mycorrhizal network. The fungus is physically linking these two different root systems. But As it grows, a few things can happen. It can be grazed, the trailing edge might die, it might be disrupted in some way, so that now that connection is broken. And if you repeat this process over and over again, what you end up with is something that is compatible with the data from the maps, excuse me, sorry, but it's not a common mycorrhizal network. So with this data, this genotype here is the same genotype here, so it spans 21 meters, but you notice in this case, none of the roots are connected. But again, this is compatible with the map. And so what this means is when we're looking at these maps, we assume that they're demonstrating a common mycorrhizal network, but in reality, they might not be a mycorrhizal network. It might be just these patches of a single genotype that is a legacy left over of the fungus growing through the forest, but it doesn't mean that everything is connected now, that all the roots are connected now. So one really outstanding question that we need to better tackle or better understand is like understanding how long those hyphal links persist for. Is it days? Is it weeks? Is it months? Like we don't have a great handle on that, because how long they persist for is really critical to then understanding what is their functional importance. If they're not lasting that long, it's, you know, are they that functionally important to the tree? And again, this is from the tree perspective, not from the fungal perspective. But probably more importantly, or further to that point, is we only have 5 of these maps done in the world, and they include only 2 tree species. So there's 4 maps done in Western Douglas fir forest in a very small region. And then one of these maps has been done in a Japanese pine forest. So we've only looked at 2 tree species, and there's an estimated over 70,000 tree species. When I look, and then only 3 ectomycorrhizal fungal species included in this. When I added up the total area mapped across these 5 studies, it was equivalent to just 1 hectare. And this is 1 hectare of over 4 billion hectares of forest worldwide. And when I say these, when I kind of, you know, give you context for these numbers, please know I'm not suggesting that we even need to map half of the forest worldwide, or even half of the tree species out there. But my argument is I think we need to know more than what we have to generalize about the structure of these common mycorrhizal networks in forests and in different forests. So again, we've only looked at 2 different forest types, so it's a bit of a stretch to know the structure of these mycorrhizal networks in other forests. But again, it's likely that the trees are connected, but which trees, how many are involved, how long those connections last, I don't think we have a very good idea yet. Okay, so updates in light of evidence. So this claim that common mycorrhizal networks are widespread in forests, we said that it was limited just because we don't have that much information on CMN structure in the field. But what we do know is that adjacent roots are often colonized by the same fungal species. And so again, we think it's very likely that these common mycorrhizal networks are expected to form, but we just don't know how widespread and how long they persist in forest to be functional with respect to the trees. Okay, so this next claim, resources are transferred through common mycorrhizal networks, resulting in increased seedling performance. So to look at this claim, what we did is we did a really wide search for any field studies that were conducted in forests or plantations. And they fall into 2 categories when we're talking about looking at CMN function. So some of them are looking at the potential effects of access to common mycorrhizal networks on seedling performance. So if seedlings, if they can plug into the network, do they do better? The claim says, yes, seedlings, you know, they benefit from plugging into the mycorrhizal network. The other aspect that's looked at, usually, well, most often separate from seedling performance, is this idea of resource transfer. Are resources being moved trees to seedlings or tree to tree through these common mycorrhizal networks? And I want to highlight that there's actually only 2 studies out there that have put those together, that examined resource transfer in the same batch of experiments that looked at the potential effects of seedling performance. Rarely do experiments put those together. And we know why they're difficult experiments to do. but it's important in the future to try to connect those ideas. So we found 26 field studies that looked at these two different aspects of CMN function. So 12 of the studies looked at resource transfer, 14 assessed seedling performance. You can see the location of these studies. So the black circles, these correspond to studies that were done in ectomycorrhizal forests, so one kind of mycorrhizal type. The blue, there's just a few dots. This corresponds to field studies that were done in arbuscular mycorrhizal forests, so another mycorrhizal type. And then the purple, there's just a couple or three of them, I think. This corresponds to field studies that were done in mixed forests. So the majority done in Western North America, but we were surprised actually when we pulled it all together that there are a number of studies that have been done worldwide. So that was encouraging. One other note about these studies is most often we don't check to see if there was a common mycorrhizal network present. We assume by how we're doing our experimental manipulations, but rarely do we check. Some studies don't even look at to see which fungi are colonizing roots of their test seedlings. And so this is just sort of a limitation to be aware of. So there's different ways that we assess the effects of common mycorrhizal networks on resource transfer or seedling performance. The first way that we do is we use what we call like natural barriers, and where we're assessing different mycorrhizal types. So I'll explain this. So this is the approach that Samard et al. Used in that first paper looking at below ground carbon transfer in the field. So this is really one of the papers that, well, it was the first controlled replicated field experiment on this topic done in the field. It was a remarkable paper. It's what got me into mycorrhizal ecology. Melanie was part of this team. And after this came out, I was a graduate student then, or an undergraduate. And I remember calling her and like basically begging her, can I please join your lab? I just thought this was just, I mean, probably like many of you, just amazing, amazing. And it was super, super groundbreaking for the time. So the way that they set up their experiment to assess carbon transfer below ground is they planted an ectomycorrhizal seedling, so in this case Douglas fir, next to another ectomycorrhizal seedling, birch, and then they used this third seedling as a control, and it formed a totally different kind of mycorrhizal type, arbuscular mycorrhizas. These two types of mycorrhizas, so ectomycorrhizas and arbuscular mycorrhizas, there are different species involved, but importantly, they're very functionally different. And so you can see with this schematic, the ectomycorrhiza fungi, these are the little purple guys in the picture, they put out a lot of hyphae, and it's really dense in the soil. And compared to the arbuscular mycorrhizal fungi, shown here in these little orange root tips, they put out less hyphae. They're better or worse at acquiring different nutrients, but there's just not as much of it in the soil compared to, say, ectomycorrhizal fungi. So the reason we're using this arbuscular mycorrhizal plant is that if we apply labeled carbon dioxide to, say, to this ectomycorrhizal plant, and then trace it down through the roots into the soil, if it ends up in this arbuscular mycorrhizal plant, the only way it could have ended up there is if that carbon was leaked from the roots, went through the soil, picked up again by another mycorrhizal root, and then traveled up the plant. So the carbon is going to move through what we call a soil pathway. And so we're trying to control for that by using this type of mycorrhizal plant. So what they found is when they added labeled carbon dioxide, some of it ended up in this ectomycorrhizal, they labeled the birch, some of it ended up in the Douglas fir. A little bit also ended up in the arbuscular mycorrhizal plant. But the limitation with this, that, you know, again, with many of these studies, when we were doing the studies 25 years ago, or even in my own research, as the field develops, you become more aware of the possible confounding effects or the possible limitations to your experiments. This happens in every field. It's not particular to mycorrhizal ecology. It's just how science develops. And so now we know a lot more about these arbuscular mycorrhizal fungi than we did 25 years ago. So what we know is that if you dropped a little bit of resource into the soil, that just based on these functional differences alone, we would expect those ectomycorrhizal fungi to take up more of that resource than the arbuscular mycorrhizal fungi, regardless of the CMN. So what this tells us is this arbuscular mycorrhizal plant, when we're using it to account for the amount of carbon or resource moving through that soil pathway, it's likely underestimating that amount. So there was already a tiny amount moving between the two ectomycorrhizal plants, and we're suggesting that maybe a lot more of it actually was through the soil. So just kind of, keeping that in mind. So this arbuscular mycorrhizal plant, or this using this natural barrier, it's a great way to try to control for what's moving through the soil pathway. And even though, as I explained, there are some limitations to using it as a control, it's still really necessary. So for example, in this paper, this came out in 2016, and it was a science paper. So as you know, I mean, it's one of our most prestigious journals. And this experiment was really neat because what they did is they applied CO2, which had a unique isotopic signature, to mature trees. So up to now, most of all the studies, they're working with seedlings, and this is the first one that's assessing the potential for mycorrhizal networks in a mature forest. So these are big, tall trees, like 30, 40 meters tall, and this took place in a Swiss forest. So they labeled the trees for 5 years, labeled one tree for 5 years, and they controlled so that the label was not dispersing in the air to the nearby trees. And what they found is some of that carbon added to this tree ended up in little amounts in nearby trees as well. But these trees, they all belong to the same mycorrhizal type. They're all ectomycorrhiza. So there is no control. So we don't know, we can't account for how much was moving through the soil. So it's a neat experiment because it was done on large trees, but there was no control. We don't know. They didn't account for how much of that carbon could have been moved through the soil pathway as opposed to moving through the common mycorrhizal network. So what this means, so what I have here is a table. In this column is the different method of separating a CMN from a non-CMN effect. And so I've gone over 2 different ways. These are just a list of the studies. I didn't put all the information about it. Over here is the resource that they were looking at to see which resource, if the resource was moving through the common mycorrhizal network. And in these columns, I'm going to pose some alternatives. So based on the data and the way that they set up these experiments, we can't rule out that carbon or nitrogen was moving through the soil pathway. So this is an alternative explanation. And I should say, so the studies presented in this table, they claimed that there were positive effects of the common mycorrhizal network on, say, resource transfer. So that's one way that we assess the effects of common mycorrhizal networks. That way of using a natural barrier, we cannot assess its effect on seedling performance. We can only use it to assess that resource transfer. So the other method that we use is and I've used experiments like this in my lab, we plant seedlings into these mesh bags, put them out in the forest, and the mesh bags, in this case, they differ in their pore size. So when the pore size is really, really tiny, it can prevent fungi and roots from growing into the bag. So like this right here. So there's still mycorrhizas in the soil, so the seedling can be mycorrhizal, but its fungi are not able to get out of the bag, and no fungi are able to get into the bag. So there's no way that the seedling can form a common mycorrhizal network. Then we'll compare it to a seedling planted in another mesh bag where the pore size is a little bit bigger so that fungi can grow in and out of the bag, but roots can't grow into that bag. And then there's usually a third treatment where a seedling is planted and it has full access to the CMN and to root interactions as well. So this type of experiment has been used to assess resource transfer, so in 2 different studies. But when you look at their results, they found that this mesh treatment actually had no significant effect on net carbon transfer. And in this study, there was actually a little bit of carbon moving below ground, even when the mesh was preventing any common mycorrhizal network from forming. So to us, that suggests that carbon is moving, and it's likely moving through the soil solution and maybe in root exudates, like kind of the liquid that sometimes roots will release. In the other experiment that used this same approach, they found, or they said, we suspect that the carbon moved predominantly via mycorrhizal soil pathway. So again, this little bit of carbon, and they were very tiny amounts of carbon moved in both cases, but it's likely moving through the soil. Okay, so I've added on here, our table is just growing. Here's a different way of assessing mycorrhizal networks from non-mycorrhizal networks effects. And we have these two studies. One was looking at them at carbon moving, and the other one is looking at carbon and nitrogen. And we would say a very plausible explanation, which is also in these papers, is that the resource was moving through the soil, not through a common mycorrhizal network. So again, as you're setting up experiments and as time passes, it often becomes more obvious about limitations to your experiment. And again, this is just a normal development in field. You look back and you look at those experiments, oh, they're really good for the time, but now we can see that there are some problems to them. So I don't want this to be not criticizing the experiments, but we're scrutinizing the interpretations of those experiments now. Okay, so with this experiment where we're planting seedlings in mesh bags with different pore sizes to manipulate access to the mycorrhizal network, the other thing that could be happening is that with those mesh bags, they might be limiting the soil volume available for foraging hyphae. So let me explain this in a different way. When you bring home a plant, you put it in a pot, you don't want to put it in a pot that's too small because it gets root bound. And then the plant doesn't do well because its roots are not accessing the nutrients and water and everything it needs because it's all bound up in that little pot. So we want to make sure that we're planting in a pot that the roots can grow. So it's the same thing with mycorrhizal fungi. When we're putting those seedlings into the mesh bags, we want to make sure that they're big enough for the roots to grow. And we haven't seen any indication that anyone uses mesh bags that are too small for the roots. But what we are concerned about, though, is those mesh bags actually may be restricting the fungal hyphae. So instead of the roots being pot bound, the hyphae are becoming pot bound. And because the hyphae are pot bound, the seedling that those hyphae belong to doesn't grow well, it doesn't survive well. So what we interpret as this seedling has no access to the common mycorrhizal network, therefore it's not doing well, we would argue, well, maybe it's just because the fungi, the hyphae can't grow out of the bag. And so they're getting bound and the seedling's not growing well. It's nothing to do with the common mycorrhizal network, but it just happens that these mesh bags are restricting hyphae to different degrees depending on the treatment. So I'll show you an example of why this is a problem. Okay, so this is from a study by Test et al. On the y-axis here, this is the percent seedling survival. And here are their different mesh treatments. So here, this is the mesh treatment where the seedlings have access to common mycorrhizal networks and they can interact with roots around them. Over here, on the other end, the seedling cannot access mycorrhizal networks and it's not interacting with surrounding roots either. So here the authors interpreted these results that when seedlings have access to mycorrhizal networks, their survival is the highest. And when they can't access these networks, survival is the lowest. So they've interpreted the results all in light of access to mycorrhizal networks. But what we're saying is that there's this potential confounding effect that in this treatment, the hyphae are able to grow and explore, access resources, get water for the seedlings. So the seedlings are doing better. And in this case, the hyphae are bound in those mesh bags and they're not able to forage for the resources that the seedling needs. And that's why the seedlings are doing worse. So again, this might not be the case, but until we eliminate this confounding factor, we can't be sure of the role of common mycorrhizal networks affecting seedling growth. So the other confounding factor with this experiment when we're using these mesh bags is that we might be changing the fungal community composition inside from outside of the bag, and how much that fungal community changes from inside and outside of the bag might change depending on the treatment. So across all the studies we looked at, only 4 actually measured fungal community, mycorrhizal fungal community composition inside and outside of the bag. And in 3 of 4 studies, they found that there was changes in the mycorrhizal fungal community composition inside, outside the bag as a function of the treatment. But the other thing that could be going on, and again, we only became aware or started thinking of this like more recently, is it's not just mycorrhizal fungi that influence seedling performance, it's also fungal pathogens. So if we are manipulating fungal pathogens in a way that confounds with these treatments, then the seedling response might be to those fungal pathogens, and again, has nothing to do with the common mycorrhizal network. So here what I've done, so these are the experiments using these mesh bags differing in pore size. Here's the list of the studies, and here's some what we think are plausible alternative explanations. So all these studies are reporting positive effects of CMNs on seedling survival or growth. And what we're saying is there's these alternative explanations. In all cases, it could be a change in pathogens, the pathogenic fungi in the soil. It could be a change of the mycorrhizal fungal community composition as a function of those mesh treatments. It could be a change in that soil volume available for foraging hyphae. And in these 2 cases, we think actually there's evidence that roots help the seedlings grow better, which is a little bit unusual. So the other way that people have addressed this problem of trying to control for the amount of soil that those foraging hyphae have to access is by using this other kind of setup. So it's similar. So we're still planting seedlings in mesh bags, but in this case, the mesh bags are of the same pore size. But to manipulate access to the mycorrhizal network, what they do instead, so the seedling is planted into a bag that's where the fungi can grow in and out, but to make sure that the seedling does not have any access to the common mycorrhizal network, they trench around the bag. So there's a lot of soil available for these fungi to grow and access, but they can't form a mycorrhizal network with seedlings or trees nearby. But anytime that we are using these mesh bags or trenching, we could be changing the fungal community composition. This is a potential confounding effect. So in these studies, this is a different approach, same thing. So we argued a plausible alternative explanation is that you're changing the fungal community, and that's what the seedlings are responding to, nothing to do with the common mycorrhizal network. Here, I've highlighted these 4 studies, and I wanted to highlight them for you because those are all studies that we worked on. These are from our labs. And so I just want to make it clear that, again, this is not an attack from the outside. This is us, when we worked on this review, I think we each had our own little moment of, oh, like, I know I described it this way in the paper in the discussion, but now I realize it could be these other things. And I remember like Melanie, when she pointed out in one of my studies that, well, you said it was from the mycorrhizal network, Justine, but don't you think it could have been from the soil pathogens? And it was that moment of just like, yeah, it totally could have. And I'm so thankful I was working with very close friends, because it was kind of in some ways very humbling and sometimes a little bit humiliating, to be honest, of just like realizing that it's not that we were wrong, but we weren't right. And yeah, so that's why I highlight this, just to note, we're not just picking on other people's studies, we're very much self-reflecting and looking at our past studies as well. So there's one experiment that was set up a little bit differently, where they planted seedlings in these mesh bags, but what they did is they overlapped, or they had layers of coarse and fine mesh, and they created it in such a way that the only way that anything could get through, a fungal hyphae, it would have to travel through this mesh. But it was a really nice design because the soil solution, nothing through the soil solution could get through the mesh, because there was this little air gap all the way around the bag. So just the fungal hyphae could get through, but that was it. And in this experiment, what they did, so this was in a pine forest, and they chopped some pine trees down, and they put sleeves on the trunks, on the leftover stems, and then they added this water with dye in it. And so then they could follow that dye, and they could track it into seedlings that were planted around the stump. And what they found in one colonized root tip of one seedling, that they found that dye. And the only way that the dye could have gone there is if it was in water, moving through a common mycorrhizal network and ended up in that seedling. So this is our strongest evidence that resources are moving through a common mycorrhizal network. But I want to stress, it's one data point in one seedling of a study that probably contained hundreds of data points. But when we look through all the studies, this was it. So, and it is something I would like to follow up on anyways. So as you're listening to me, you might be thinking, maybe you don't believe my list of confounding effects, or you're not convinced by them. And I would say, okay, that's okay. But even if you ignore all those confounding effects that I just talked about, the story still does not hold. So this idea that seedlings, they benefit from the common mycorrhizal, from plugging into these common mycorrhizal networks. What we found is that sometimes seedlings did better, sometimes they did worse, but most of the time there was no response. And how they were influenced by potential access to these common mycorrhizal networks, it varied by which tree species was involved in the experiment, what kind of soil, the distance between mature trees and seedlings, the extent of tree mortality, duration of the interaction, how long the experiment was set up, and the neighborhood, the forest neighborhood. So there is no way to generalize in any way to say that seedlings do better with common mycorrhizal networks. There's a lot of variation that's going on here that we don't understand, that we need to understand before we can start understanding how this interaction works. One thing that we did find, though, is that only 5 of 28 experiments, so this is only 18%, showed positive CMN effects on seedling performance. So again, by far, the majority of experiments, seedlings, they showed no response if they had access or not to mycorrhizal networks. The one thing that we did find, though, was the importance of root competition. Seedlings tend to do a lot worse when they have access, when there are access of neighboring roots. That came out kind of loud and strong. So this claim, so resources are transferred through CMNs, resulting in increased seedling performance. We said there was no consensus, because with these field experiments, there's all these confounding effects. There's alternative explanations available that don't involve common mycorrhizal networks. And until we eliminate those, we can't be sure of the role of the common mycorrhizal network. But what we do know, which we thought actually was kind of interesting, is that resources, they are moving below ground. And in some cases, there's no mycorrhizal network that needs to be involved. The small amount of nutrients or resources is trickling between different seedlings or trees in the absence of a mycorrhizal network. And then as I mentioned, so there's approximately equal evidence that connecting to a putative CMN improves or inhibits seedling performance, but neutral effects are most common. Okay, so this last claim, mature trees preferentially send resources or warning signals to their young or kin through CMNs. And this claim has got a lot of traction. And so this is an example. So this was a one-page infographic put out in the National Geographic from 2018. I know you can't read all the little text and stuff like that. So it says talking trees, has an image of an old growth forest. It talks about how the mycorrhizal network is moving these resources to different species, changes depending on season. Over here, there's a tree that's been attacked by some worm or something, and it's sending warning calls to a nearby tree. That's kind of the gist of this infographic. We were really surprised to find that there are no peer-reviewed published field studies testing this claim. So then we wondered, so where is this coming from? And what we found, so we really, there was no stone left unturned when we were doing this this review. Obviously we looked at published papers, but we also went to graduate theses as well, and then looked at them to, where is this claim coming from? And what we found, so we did find one graduate thesis, and it was a field experiment, and actually it was a really neat field experiment where this student, so it was in a Douglas fir forest, and they were interested in testing the influence of kin, so whether trees and seedlings were related or not, and common mycorrhizal networks and how they affected seedling survival. So they took seed from a faraway stand, so with the assumption that that seed would be unrelated, not kin, and then seed from the focal stand, again with the assumption that that would be related or kin. And then they sowed the seed besides mature trees, and then they manipulated access to the mycorrhizal network with those mesh bags. And so what they found, I'm going to walk you through this graph and sort of do it in parts. So the first thing, okay, so on the y-axis, it's called an odds ratio of seedling survival. All you need to know is when this number is above 1, the seedling survival increases. When it drops below 1, it decreases. So this dark colored bar, this corresponds to seedlings that are non-kin. So they're unrelated to the tree that they were planted by. And what this student found was actually non-kin seedlings survived, they had higher survival than kin seedlings. So this is opposite to what we're hearing. When they looked at the effect of mycorrhizal networks, so manipulating it through these mesh bags, they found no effect. So this, you can see there's no bars because there was no effect at all of whether the seedling had access to the mycorrhizal network or not, had no effect on seedling survival. And this is looking at an interaction, which is saying a similar thing. Not surprisingly, the one thing that did affect how seedlings survive was the site that they were planted in. So this site survival was really high. These sites survival was really poor. But again, so this is counter to the story. We did come across 2 greenhouse experiments where they're also looking at this question. So one of the greenhouse experiment, it was looking at this idea of kin and carbon transfer. So they set it up where they had 4 different families. And what they found is in half the families that this we'll call the donor seedling and the receiver seedling, they're both Douglas fir. In half the families, the donor seedlings preferentially directed carbon to its kin. In the other half of the families, they didn't see anything. But importantly though, this happened in the absence of common mycorrhizal networks. So they're using mesh again. And what they found is that, so this carbon was transferred between the seedlings in the absence of a direct linkage between the mycorrhizal hyphae. So again, small amounts of carbon moving below ground, but not through the mycorrhizal network. There was one other experiment we found. So again, this is done in a greenhouse where they planted Douglas fir and ponderosa pine seedlings together in one pot. They then used mesh to manipulate mycorrhizal networks. So they had one treatment where the seedlings could form mycorrhizal networks, but their roots didn't interact. They had another treatment where the seedlings could form mycorrhizal networks, but the roots can interact. Sorry, I think I said the other one wrong. The first one, form mycorrhizal networks and the neighboring roots could interact. And then they had a last treatment here where the seedlings couldn't form any mycorrhizal networks with each other and their roots didn't interact. And then what they did is on the Douglas fir, they put some spruce budworm on the Douglas fir and the budworm eats the needles. And what they found, and this is really interesting, is that when the seedlings could form a mycorrhizal network, but the roots didn't interact, that the defense enzyme expression in this ponderosa pine increased. So this Douglas fir is getting attacked and this seedling is responding. And it seems so something has to be happening through the mycorrhizal network in the absence of roots. But in this treatment, the treatment where the common mycorrhizal network can form and the roots of the seedlings can interact, the effect it disappeared. It was totally canceled out. And the important thing to remember here is that common mycorrhizal networks in the forest, they don't occur in the absence of roots. They can't, unless you're talking about a very, very small scale. But a common mycorrhizal network, it is the physical connection of roots with this fungal hyphae. So we think this experiment is interesting, and I have this little picture here that they showed similar results when they cut the needles as they did when they added the worm to it. But the relevance to a forest, we're not sure of. And probably the most important point is that we don't think that you can use results from a greenhouse study and extrapolate it to a forest. So 2 seedlings growing in a pot in a greenhouse is very, very different than what is going on in the forest. And so again, we haven't seen any peer-reviewed published studies testing whether this signaling is happening in a forest. Yet, when you hear the story, it is often told in the context of an old growth forest, big trees talking to each other. And yeah, we would just say that this is a massive stretch. So for this claim, the mature trees preferentially send resources to each other, warning signals, we found no support. And so whether this happens, it remains to be demonstrated. Okay, so the other part of our study is we wanted to look at how scientists were interpreting claims about the Wood Wide Web. Is it possible that we scientists are shaping the popular narrative? It's very easy to kind of wag our fingers at the media and the public and their poor understanding of science, but what role have we played in this? Okay, so the way that we did this is we selected what we called were influential studies in common mycorrhizal network research. So these are ones that have, at a certain date, May last year, they had received over 50 citations, which in my world is quite a few citations. And so we called those the influential studies. And then we looked at all the papers that had cited those influential studies to see how they cited them. And we would categorize the citation as either unsupported or supported. And I'll give you an example just so that's clear in your mind. So here's an example of an influential study. We talked about this one at the beginning of the talk. This was by Byler et al. It was that beautiful map, the map of common mycorrhizal network structure. And we agreed with their conclusion from this paper that single fungal genets colonize multiple trees of the same species. Some trees are colonized by multiple genets. Yes. When we looked at papers citing this influential study, we then assessed, okay, so how are they talking about this study? And what we found is that in about half of the time, people were citing this, for example, for evidence of carbon transfer. So Byler et al., they never looked at carbon transfer in this study. They made a map, and it was a great map, but they never assessed resource transfer. But yet, many papers are citing this influential study as an example for resource transfer. So that would be an example we would say that's an unsupported statement. So what we found, so this is for influential studies on CMN function. So this is looking at the studies that assessed resource transfer or the effect of CMNs on seedling performance. And we tracked it through time. So what you're looking at on this y-axis is the percent unsupported citations. This is time. And over here is the total number of citations. So this gray cloud behind all the points, this just represents how many citations per year in total. So you can see, not surprisingly, that the number of citations has increased in time. What is kind of interesting, though, is how much they've gone up in the past few years. Importantly, what we found, though, is that in 1997, so this was the year that one of those influential studies was first cited, that about 20% of the papers citing one of those influential studies was making an unsupported statement. Now, 45% of the papers citing those original studies are making unsupported statements. It's quite a lot. When we looked at CMN structures, so then these were our papers, original papers looking at the mapping, the structure of the common mycorrhizal networks. The first citation came out in 1999. In general, there was a very low rate of unsupported statements made in these citations. Only 2% is probably where we would want to see it, but now it's up to 25%. Not as high as with the CMN function studies, but still quite a bit. And this is interesting because the total number of citations per year has actually gone down for this batch of studies, but the percent of unsupported statements has gone up. So then we looked at the nature of those unsupported citations. So, what were they saying? Could we generalize in some way? And we found that there was bias that promotes the positive effects of CMNs in forests. So bias being, so exaggerating the presence of CMNs in forests, overlook alternative explanations in favor of positive effects, inappropriate extrapolation that inflates the CMN effects in forests. And I want to be clear that it was very disappointing, but all three of us made that naughty list as well. When we went back and looked at our papers and looked at how we were citing some of those original papers, it hurt. It hurt to see that we were making sometimes, unsupported statements. And so what this means is that we scientists have also become vectors for unsubstantiated claims. So some people, when they hear this, they'll say, is it such a big deal? Because this idea that trees talk to each other through below ground fungi, it spurs a lot of social action for people to care about forests, to care about fungi, to get interested. And everybody might land in different places. We can talk about this after the talk. But for me, this is where I've landed. And so this is a quote by some authors, Cope and Allison, from a 2010 paper. And I have it written down because I can never say it as well and as nicely as they do. So I'm just going to read it out. So scientists have as a discipline our own code of conduct. Central to it is a commitment to faithful reporting, to acknowledging our study's limitations, to evaluating bodies of evidence without selectively excluding information on the basis of its desirability. In short, a commitment to truthfulness. So the demonization of some things and sanctification of others, in this case, the Wood Wide Web, though perhaps helpful in spurring social action, may be more harmful to us in the long run by giving unconscious permission to breach that code and thereby eroding the foundation of the scientific discipline. So with the Wood Wide Web, I think for many of us, we liked where it went. We liked the story of cooperation in forests, trees helping one another. And so it might have been, like, unconsciously or not, kind of giving it a free pass in terms of, like, we like where it's going. And so, do you turn a blind eye, or is there confirmation bias going on there that we like this story? And so the evidence is getting distorted. But imagine another case where people were painting a picture of a forest in a way that was like, oh, forests have no value, fungi have no value. I am sure that there'll be a lot of scientists then that would then call that out and say, how do you know that? What's your evidence? So in that case, we are very uptight about how science gets distorted, but we need to hold everything to the same bar. If science gets distorted, it doesn't matter if we like where it goes or not. We need to be calling it out if we want the public to trust the scientific enterprise. So moving forward, so hopefully, well, not hopefully, I'm convinced that this popularized story, it doesn't stand up to scrutiny. And just to let you know, so kind of where we're at a little bit. So to date, 99 news stories from 91 outlets worldwide have covered our paper. And so we're happy that the message is getting out. This doesn't mean we have no control in how people interpret our paper or what they're thinking of it, but I am happy that the message has been widespread. We know that there's bias in the scientific literature. Again, so we've demonstrated that this is a problem. And I actually don't think that it's just in mycorrhizal ecology. I think it's happening in other fields as well. And again, we can talk about that later. But I think what's really important is that we need better scholarship. So the citation analysis, it demonstrated that there's a problem. We have some bias, but it's more than that. So for example, this book, Finding the Mother Tree, it's a memoir. It wasn't peer reviewed, it's a memoir. And Suzanne, she very clearly states in the back that, her memories are fuzzy sometimes. It's a memoir. But since 2021, 21 articles, so in Science and Humanities, they're citing this book as evidence for common mycorrhizal network structure and function. So again, this is not peer-reviewed material, but yet people are now citing this book as evidence. So moving forward, in our paper, we spend some time, we want to be forward thinking. We do not want to put a chill on this field of research. If anything, we're hoping that our paper will reset and kind of reorient the field a little bit. And we're really excited about the next generation of experiments and thinking about how we can eliminate some of these confounding factors, and just thinking about them and acknowledging in our work that there are some limitations, and being more careful when we cite the work of others or going ahead. And if we don't do this, my concern is that the Wood Wide Web will become a fantasy beneath our feet if we don't get our house in order here. And just to end it, so this comes up sometimes, it's come up in some of the major newspapers in the comment sections. I didn't have the heart to go through all the comments, but Jason, my co-author, he has spent some time combing through those comments, very brave. And one thing that's come up is that people think that, oh, we must be funded by big timber or big forestry. And I just want to be clear, there was no external funding to this project. It was really something that we worked on kind of the side of our desks. And of course, our respective universities paid our salaries, but that was really it. So with that, I am done talking, and I'd be really happy to take some questions.
Amazing talk. Thank you so much, Justine. The first question that we had was about external funding, and I think that has been addressed. So
okay, yeah, I mean,
yeah, I mean, unless there's anything else that you'd like to say about it, I think it was well addressed there. The first question then is Jim Watkins. Jim, do you want to unmute yourself and ask Justine your question?
Hold on, I'm trying to find my first question because I had
you had several. It's up near the top at 7:26 p.m.
7:26. Yes. I think you actually showed that this wasn't the case. When you said that the root connection, well, the connection between the fungus and the roots got severed. And then I was looking in some of your other pictures. I was just questioning, how that could happen, because I would think that there would be literally probably hundreds of connections with the fungus to any single root system.
like with the trenching study, is that what you're thinking of? Like where they?
Well, I think at the beginning you said how that you thought in the forest what could happen is, the connection got severed.
Yes.
And I was thinking, how does that, if it's just one connection, it would be easy to sever, but I would think there would be hundreds of them.
Yes, no, and that's a good point. So the other thing to remember is we often, so with the common mycorrhizal network mapping, is that we've only focused on a couple species, like Rhizopogon in one, and I can't remember the other fungal species, another one off the top of my head, but only 2 species. But it's possible and likely that, there's all these other mycorrhizal fungi in the system that can be part of this common mycorrhizal network. So this idea that it's just like one fungal species that's holding all the trees together is, it's probably unlikely. And to your question, there could be other fungal species involved in the common mycorrhizal network, and maybe they're patchier. And that's the thing, it's like we think about these fungi as these like massive things underground, but depending on the species, and as you probably know, some of them are really patchy, they just form small patches. Some of them might form big patches. And we just don't have a good handle on what this network looks like. And you're right, so one connection is broken here, but there's still these connections over here. And so is that functionally relevant to the tree? And yeah, I take your point, and I would just say that we don't have a good idea of what the mycorrhizal network looks like, which species are involved, how long those links last. So there's still lots to discover there. Yeah.
Do you think, this is a follow-up, do you think now, based on what you went through as an experience, that we can tend to do as humans in general? I think we tend to want to, when we find something new, we tend to want to, we get excited about it. We tend to think that that can be the only possible view.
Yes.
And sometimes that view also is kind of anthropomorphized,
Yes.
so we're kind of taking our human worldview and imposing it upon nature. So, obviously we use a internet, and so we found something, similar.
Mhm.
Looks like we found something similar. So we get excited about it and we forget that there could be these other possibilities.
I think that's a lot of what happened. Even I would, I'm only going to speak on my behalf, but so the field experiment I set up looking at common mycorrhizal networks, we were so focused on like, what's the role of the common mycorrhizal network? And it's like, we didn't even consider that there could be other things at play. And I think for a lot of people, like we're really interested and excited about common mycorrhizal networks. And it's like, for me, and again, I'm not going to speak on anyone else's, but it's like I didn't think about the alternative mechanisms and I didn't test for them. And that's a problem. You're right, because it was, we were so excited about it. And the other thing too, it's like we're so quickly to anthropomorphize things to make it more understandable, I think. But the problem with that though is when we have this zoocentric view of plants or fungi, it puts up blinders, I think, to understanding them for what they are. So the more that we try to map on the internet or like human behaviors onto fungi and plants, I think it's possible the less and less we're going to see and understand about them, because they're totally different organisms. And whoever is clever and brilliant enough, I think, to approach trees and fungi for what they are, I think that they are going to make amazing discoveries. But me personally, I think it's a bit of a trap to keep anthropomorphizing and keep wanting to map on zoocentric mechanisms and behaviors onto organisms that are totally different than animals and humans. Thank you for the question.
Someone identified just as Zoom user had a question about saprotrophic networks. Do you want to unmute yourself if you're still here?
Yes, I was simply asking about whether armillaria might actually be playing a role here, or other pathogenic fungi, if they might be stealing resources from one and passing to another, or
maybe, like, I think though, like trees, they have things in place so that like if they're losing resources, it's obviously not of their volition, and sorry to use like a very human-centric term, but the role of saprotrophic fungi in these networks, and if they're acting as a network, I don't know. It's a very good question. I do know from my own research, so where we set up our study looking at the role of common mycorrhizal networks, and we set them up in a series of pine stands. Some of them were healthy, intact stands, and then all the way to stands where the trees had died. All the trees had died from mountain pine beetle attack, so they were standing dead. And what we found in the soil of these beetle-killed stands, there was a lot of pathogenic fungi. So when we set up our experiment to manipulate common mycorrhizal networks, what we found is that seedlings that were in those mesh bags where the fungi couldn't grow in or out, that they did better than seedlings that were in mesh bags where the fungi can grow in and out. We initially interpreted as, oh, when the seedlings have access to mycorrhizal networks, they're actually doing worse. But what I think it is now is that those seedlings that were in the mesh bags where fungi couldn't grow in and out they were actually protected from pathogens. And that's why they were growing better. And actually, that was one of my moments when that was what Melanie figured out in my experiment that I had never thought of. And I think that is probably the more likely interpretation. And now I think it is a really important thing to be looking at pathogens. And again, this is something, we weren't thinking about so much 25 years ago. I think 25 years ago, and I know there's some people on Zoom that could help me out on this. I think people like mycorrhizal ecologists, we were really working hard to say, no, they're not pathogens. They're not bad. They're not harming the seedling. They're doing something good. And so we were kind of, trying to separate ourselves from pathogens. And then now 25 years later, I think we recognize that, hey, pathogens are part of the mix. And we know that mycorrhizal fungi are not pathogens, but they occur in this mix of soil that includes pathogens. And we can't
Right.
ignore them anymore. Yeah.
Right. And how about mycoviruses? Now that they've found that mycoviruses exist in truffles, for example, or
Yeah.
they exist in other systems where you've got different species like salal and Douglas fir.
Yeah.
Could you have a virus that's transmitting from one to another?
I don't know. That's such a good question. I have no idea. And I think, it would be really interesting to look at it. And I think that's what I've sort of landed on too. It's like, for so long, we've been thinking about these networks or transfer as like helping the trees. And it's like this cooperative unit. But why, why would they just be transferring resources? Like, could they not be evolved and working as like spreading viruses? And I think that's a really interesting question. And I don't know what the answer is though. Yeah.
Okay, it would be, I just think it might be an interesting thing to look at is maybe they might be vaccinating each other too, that way. Making, if you spread a hypovirus or a hypovirulent virus from one tree to another, they might actually be passing immunity to each other as well. So you could have both the negative of making each other sick, or you could have the positive of inoculation.
Yeah. It's a great point. Yeah. Thank you.
Yet another column to add to your possible confounders, right?
I was like, I need to make a little note of that one. Yeah.
Paul Sadowski, you had a question.
Yeah, hi, I enjoyed it very much. Could you just go into greater detail on what constitutes a soil pathway?
Oh, yes. Yeah. And sorry, it's always, it's hard to give a clear image of that. Okay. So if you imagine, you have a root and it's colonized by mycorrhizal fungi, and then a root over here, it's colonized by mycorrhizal fungi. The soil pathway represents, so say if a resource, it's leaving this root, it's then leaving the mycorrhizal fungi, entering into the soil, and say it's carbon, some kind of molecule that contains carbon. And then over here, a short distance away, is another mycorrhizal root. And so that carbon then is leaving one mycorrhizal root, entering the soil, and then probably through some kind of solution or something, is then ending in this other mycorrhizal root. The point being though, is that this resource, so in a common mycorrhizal network, the network would form like a tube between the two mycorrhizal roots. So that resource is protected. It's traveling in a tube of fungi, basically. And so it's not, it's protected from microbes and everything else that's going on in the soil. It's just moving in this fungal tissue. The soil pathway, mycorrhizal roots are probably still involved because most tree roots are all mycorrhizal, but it's not moving through that fungal tube. It's like going through the soil pathway and then ending up in the other mycorrhizal root. Is that clear?
Yeah, so you're saying it's important that there be some sort of aqueous or other kind of solution to conduct the molecules from one place to. Okay, thank you.
Ji Hao Wang, you had a comment, question related to this topic. Maybe you'd like to unmute yourself and ask. No, see how is still here, but maybe I'll ask or I'll read his comment, and if you unmute yourself, you can jump in. The claim of CMN also downplays the importance of plant exudates, soil pathway, bacteria, microfauna, and other soil ecological entities in the transfer of nutrients.
It could. I mean, if anything, I think that the evidence we looked at, it shows that there's something moving through root exudates. But it's interesting though, because if it's moving through root exudates, why isn't it taken up by all the microbes and everything all around it right away? Like carbon it's a very limiting nutrient in the soil. Everybody wants it. And so it is really interesting that this small amount of carbon is moving through that soil solution, maybe through root exudates or something else, and then ending up in another root. And how that happens and why, I'm not sure. Yeah.
Well, someone, iPhone, you've had your hand up for a while. Would you like to unmute yourself and ask your question? I don't see a name associated, and you don't have a comment in the chat, but I see your hand is up. If you would like to unmute yourself, go ahead. And in the meantime, I will go on. Lots of interesting comments. Here's a question from Mateo Pace. Would you like to unmute yourself and ask your question, Mateo? Mateo?
He says he's got a bad connection in the chat.
Oh, okay. I will read your question. Thank you for a wonderful presentation. I loved it through and through. As a researcher working in a totally different field in the humanities, I'm wondering whether your claims on citation bias have any repercussions on the whole peer review system. I see in my own field how hot papers books are often cited, but seldom read. What is your take on the way we make and distribute scientific research? Shouldn't peer review be the first stage to spot the citation biases that you investigated in your review? And if it's not doing what it's supposed to do, what then?
Yeah, that is such a great question. So obviously this is a problem. And so we gave some suggestions in like the support, like the appendix of the paper, and some of them are really basic about just good citation practices, and as supervisors training our students, good citation practices. But, you know, and some of it too is, I think, basic stuff, and I'm guilty of it as well. Like, again, I've learned many lessons from working on this review, but, you know, sometimes you've read a paper and you read it a few years ago and you think you know what it's about. And so you're writing your paper and like, wait, I'm going to cite that paper, and you don't look at it again. Or in the case, you know, maybe you just read the abstract and you don't read the whole paper when you probably should have. So these are things that, yes, we need better citation practices. For the peer review, it is, I mean, so for me, when I get a manuscript, I'll usually check the citations in the first 2 paragraphs and just see like, okay, yeah, that's what I think that paper's saying. That's my understanding. but I have to admit, I don't check the whole paper. I'm relying on the authors to be doing that job. And, you know, and this is the thing, like so much of science, so much of research, not just science, so much of research, we are trusting each other. And I think for the most part, we can trust each other, but I guess well, I should back up. I'm a little bit more skeptical now because, and I think, you know, with students, assistant professors, us included, there's so much pressure on us to be putting out papers quickly. And I think that science is really starting to suffer from this now. And, you know, maybe for a while we could kind of get away with that model. But I think that, you know, for example, the citation bias problem, this is doing a disservice to science. And, you know, very clearly there's some bias, and what happens is, so the field, you know, for the past 25 years, we've been focused on CMNs and they have all these positive benefits in forests. And, you know, until now, until the claims got so extraordinary, did we pause and like, is this true? And so this is it's a big problem. And how to fix it, like I said, you know, I think it starts small with every day when we're training students, when we're writing the papers ourselves. But again, this is not just happening in mycorrhizal ecology. It sounds like it's happening in your field as well. And so at some point, the pressure and the amount of information coming out and the quickness of the papers, the rapid it's not helping anymore, I think. I think that something is going to happen very quickly. Or for me, like I am very concerned that the public is losing their faith in science. And right now it seems like it's kind of for a good reason. If we're having these problems with bias, we're having these myths of the Wood Wide Web out there that, you know, are only starting to kind of be reined in now. Like I worry too. I mean, and it goes further than that. Like, for example, my son, who's in grade 9 now, but when we started this paper, he came home and he said something to the effect of like, Mom, guess what? We learned that trees talk to each other through fungal networks. And that was, I think, one of the moments that I was like, oh my God, like, this is everywhere. Like, this is not just in my field where it's kind of gotten away from us. It is everywhere. And so, but on the other side, though, I think that there is evidence that, you know, science still has this very strong self-correction mechanism that when the claims get really out of hand, things start to kick in and scientists are like, okay, whoa. And so that is happening, but I guess I just wish it would have happened a little bit earlier. And this just happens to be an unusual case where it's so out in the public. Everybody knows about the Wood Wide Web, and now it's kind of all crashing down in a very public way. Yeah.
Tom Horton, why don't you unmute yourself and ask your question?
I think I did. Can you hear me?
Yeah.
Yep.
First of all, great paper and great talk, Justine. Really loved it.
Yeah.
And yeah, it's a problem. I guess what I was wondering in a scientific question is, have you seen any evidence of what form of carbon moves from fungus to plant?
This is a good question. So, like from what I understand, so I've been talking to another group who are way more focused on the physiology of like, how can this actually happen? And they are not convinced that any kind of form of sugar could be moving back to the plant from the fungus. They're just like, it's not physically possible. So then I guess the question is that carbon attached to something else, some kind of molecule that could pass up to the plant? But, you know, I would still just put the brakes on that conversation a little bit. So as part of our paper, so you read the paper, when we was going through review, the reviewers really pushed us to look at lab experiments as well. Because at first we didn't. And they said, no, you can't just look at field experiments. That's remiss. You can't just look at half the evidence. Go look at the lab experiments. So we did. And shockingly, you know, there was only, I think, so 3 experiments that we came across that showed carbon moving through the mycorrhizal network. And I think only 1 that showed that the carbon actually ended up in the plant. And this isn't, you know, lab experiment. Right. The other experiments, there's confounding effects that it could be all these other things besides the mycorrhizal network. So there's only 1. And so for me, if we're really going to start tackling this question, some of those older experiments need to be repeated. And so I don't think we want to be chasing something that's like, first, let's get a little bit more evidence that it actually ends up in the plant. You know, and this would not be field experiments. These would have to be very controlled experiments just to get that question, just to get that answer. And then, yeah, and then if it does, how, what kind of molecule, like what is that carbon attached to that it's actually moving up in the plant?
So you don't think it's an amino acid like glycine or glutamate?
Yeah. I think that's exactly it. Like
it's a source of nitrogen, and with it, the carbon
yes,
moves, right?
Exactly. It's something attached to nitrogen, and then maybe that's how it's moving. But, you know, and the other thing too, like the other thing I want to be careful about is that, you know, we're really focused on carbon in our story. We're like, you know, and there's really not much carbon moving around, and we really don't know if it influences seedling performance. But the other thing though to keep in mind is if it was like a hormone or something like that, you only would need a little tiny amount to actually influence a plant. And so, so I think it is really important to figure out what is that molecule, and I'm sure that there's tools to do that now, to try to understand the physiology behind this, like what is going on. Yeah,
Thanks.
Yeah, thanks for the question. It was good.
Greg Marley, do you want to unmute yourself and ask your question?
Yes, thank you. And most of my question may have been answered by Tom's, because I was curious, you know, the carbon that is moving through the soil matrix. Again, how large a molecule can that be that can be taken up by a root? So it's almost like another myth that this carbon is moving through the soil matrix, but
yeah, I don't know. And that's exactly those sorts of questions we need to answer. And the other thing to me is like I said, those microbes are hungry for carbon. So if there's carbon moving through the soil matrix, why isn't the microbes all getting it? How is it even ending up in another plant?
yeah.
But I don't know. Yeah. If anything, yeah, so that raised a lot of interest in questions in us to like, what is going on there? Yeah.
Other than that, so much appreciate this talk.
Thank you.
So
Yeah.
makes my brain tickle.
Thank you very much for coming.
Jim, I don't see your last name. You're just showing up as Jim's iPhone. You would have a question about forestry practices. Would you like to unmute yourself and ask your question? I'll read your question. Bottom line, what forestry practices do you recommend based on your research? The CMN angle was an important counter to the conventional clear-cutting mindset.
Yeah, no, this is a great question. So I would say that we know enough, there is enough studies out there to point the way and the direction in terms of how we could be changing forest harvesting practices without even going to this trees talking issue, like whether, you know, what the roles are of CMNs or not. We could even just put that whole thing to the side. And there's been enough research to show the importance of remnant patches, the importance of retention of trees, the importance of, just maintaining reserves out there. Like there is so much out there already that can point us the way in terms of what we should be doing with forests, that I think that the CMN thing so right now I would not suggest that any policy or management is informed by what we know or don't know of CMNs. It's too scant, it's too unsettled to bring that into the conversation. All I would say though is that there is lots done already to point the way. And even with mycorrhizal fungi, like with those fungi, there's been lots of studies to show that when we're talking about endemic fungal species, how important it is to have those reserves in the landscape. You know, there's some fungi that are cosmopolitan and will easily disperse in. But if we're interested in keeping those endemic species, then we need to be thinking about, you know, forestry practices in a different way. So yeah, to me, there's an intellectual and academic reason to figure out what these CMNs are doing or not doing. But we don't need to wait for that to catch up to move ahead in terms of how we want forests to be managed or how we think they should be managed differently.
David Hibbett, would you like to unmute yourself and ask your question?
Sure. Which one?
Oh, well, you had a question about the response to the paper.
Oh, yeah. So I think this is a really fantastic paper and great presentation. Thank you so much. I'm really curious to know what the reaction has been in the general public, because this is such a, as you say, it's part of our culture now.
Yeah.
And I guess I'm wondering if you're getting any hate mail, and I sure hope not, because I think what you've done is really, really wonderful and important. But I'd like to know, I'd like to hear about that. Thank you.
Yeah, thanks you for the question. So I would say from scientists that have reached out to me, it's been overwhelmingly positive. And these are only the scientists that have reached out to me. So it's not to, that's a totally biased sample right there. But the people who have reached out to me, have been very positive. The public has been very, very mixed. Some of them, yeah, so yes, I have received some hate mail. And yeah, and I won't lie, it's hard to take. But when I look, so I mean, as I mentioned, Jason, he went through all the comments very carefully, and he summarized them for us, and they seem to fall in a few different areas. So there is a strong component that feels like, it doesn't matter what science says. We just know this to be true. We just know that trees are communicating in ways that we don't understand, and it doesn't matter what they're saying. And so there's sort of that. They want to believe what they want to believe because, I would say it feels good. It's a really nice story. There's a contingent that are, I think, are very suspicious because of the funding angle. And all I can say is I hope I've, allayed anybody's concerns about that we're not funded from outside sources. This was really a curiosity driven of like, these claims are extraordinary. What is going on? So there's people in that camp. There are people in the public that I do think that they do recognize this is the process of science. This is what happens in terms of like interpretations are overthrown, they're reevaluated. This is just normal stuff. But again, it's just unusual because there's been so much connection to the public and so many big stories about it. And what are the other, those are kind of the major things. And then, yeah, and then just, some really nasty comments, but yeah, I would say the public is really, really mixed, and we kind of expected that. And I don't know, I'm not sure, like I have my email out there. I've actually gone into those comment sections a couple times, hello, I'm Justine. I'm happy to answer your questions and clarify any points. And I just would say that backfired really badly. People were very nasty. And so I quickly, I couldn't take that. And so I'm not doing that again. Yeah.
David Hibbett, you have a interesting question about ghost pipes, monotropa. Do you want to?
Okay, yeah, sorry, I'm not trying to hog this thing, but well, first of all, I'm so sorry to hear that. And I think everybody watching this is probably pretty horrified as well. And, there is a huge community of scientists and citizens in general that think that what you're doing is really important and really support you and people who do that sort of work. So onto a more less emotional thing. What about mycoheterotrophs? I mean, like,
yes,
ghost pipes, because we know that interplant transfer of nutrients via fungal intermediates is possible because they demonstrate that. I'm not up on that literature. I don't know what we know about the mechanisms, but what about that?
Yeah, okay, I got schooled on that recently. So first of all, the reason we didn't talk about the mycohets in our paper is because we felt like the popular claims, they don't even tackle them. It's always, it's based on trees and seedlings and mycorrhizal networks. So there's only one line in our paper that is basically giving a nod, like carbon transfer happens in this situation, in the one that you described. And then we started thinking about it a little bit more, because it's like, well, why is that system different? And so the same group of researchers that I've been chatting to, but more of the nitty-gritty physiology and how does carbon move, they explained it to me in a way that with the mycoheads, the fungi and their roots, the roots, how does this work? The roots attract the fungi, and then the roots then are, how was it? And I might get this wrong, so don't totally quote me on this, but it's like they're working the fungi then to squeeze the carbon out of them, like whether they're degrading the fungi or something else, but they're literally stealing the carbon from the fungi. And so it is like a very different situation, that it's not necessarily, this mutualistic thing. It's very much a situation of the plant's roots stealing the fungi or stealing the carbon from the fungi. And so in the compounds and stuff, because it sounds like it's a totally different mechanism, it might be all sugars in that, because it does seem to be a very different mechanism in terms of how the plants are getting the carbon compared to like trees and seedlings. But who's been talking? So Marc-André Salas, who you probably know, they've been chatting about this a lot. And so I'm learning about mycoheads, but I am not an expert on it in terms of how that happens. But I do know that they're an excellent example of carbon moving through mycorrhizal networks and ending up in plants. No doubt about it. Yeah.
Gary Gilbert, you had a question.
Hi, thanks for a fantastic talk. I, for one, am not ready to bury the shovel on Suzanne Simard's work. My takeaway from what you're saying is that there just needs to be a heck of a lot more research done. And there's a lot of players in the soil, and the techniques you're describing sound incredibly tedious and hard to execute. So for me, the jury's out. I'm not ready to, dump on it. But my question was, what could you say was correct about Simard's work? She seemed to be having carbon tracing gases she seemed to be proving that they did go from a tree to another tree. How true is that?
Yeah, so for example, my co-author Melanie Jones was involved in a lot of that work as well. And what that work shows is definitely there's carbon moving below ground, but it's not conclusive that it's moving through that mycorrhizal network. And so that's really the sticky point. We know that there's carbon moving below ground, but right now, and if anything, I mean, and I want to back up and say, just to be very clear on everybody, this is not an attack on Suzanne Simard's work at all. And so when we did our literature search, we based it on topic, and it was a very huge net that we cast in terms of getting different studies. And so, and Suzanne's work made up some of those studies, but only about 20%. So we're definitely looking at a body of research and not focusing on one person. But in some of those experiments, they very clearly show that resource is moving, and you don't need a mycorrhizal network there. Like it's just moving through the soil. And I think the interesting part is we've been sort of talking about this. It's like, how does that happen? Why does that happen? What are the players? And is it important to the seedlings and trees involved in that? And that's kind of one of the more interesting things I think that came out from the study is like, in different research, not just Suzanne's lab, we've seen it in other experiments from other groups. There's below ground carbon transfer, but no mycorrhizal networks involved. Yeah.
Thank you.
Yeah, thanks.
And maybe a final question from Colleen. You have your hand up. Would you like to unmute yourself?
Yes, thank you so much. Thank you, Justine, for this talk. This was so delightful. And I am really interested in the part of the talk where you talked about who shapes the popular narrative and what role scientists play in that. Mm-hmm. And I understand that you looked at all the papers that had cited influential studies to see how they had cited those studies. And then you categorize those citations as either supported or unsupported. And I'm interested in whether any of the papers you looked at were kind of in a gray area, like how you decided on, just those categories, I guess.
Yeah, no, that, I mean, in our appendix, we're very clear that that citation analysis is pretty subjective. And so what we did is in the supporting information for every citation that we said that we deemed unsupported, in the back of the supplementary information, we explain why. So there's like 150 pages of like, here's why we thought it was unsupported. But the other thing to keep in mind is, as I mentioned, as the field matured, when we look back at some of these experiments, there's these alternative explanations that might not have been clear when the experiment was done, but now are obvious. And so when we went back and looked at those experiments that, and some of them in the discussions, they were very clear, like, oh, this could be happening, or this could be happening. This, these are other things besides the mycorrhizal network. And that information in the discussion wasn't always transferred, though, in a subsequent citation. And so then when you would look at the citation, it would be much more, the wording would be stronger in terms of like, oh, carbon was moving through the mycorrhizal network. When the original study in the discussion, sometimes they would say, well, we're not sure, but we think it is, but we're not sure. But how it gets cited then would be stronger wording. And so that would be an example of like, when the person of whoever was making that citation, and if you're using more like definitive language, like carbon was moved through the network, then we would count that as an unsupported statement, because it's like, well, no, there's all these other pathways that are not accounted for. So definitely that citation analysis, it is subjective. And we're very clear to say when we're talking about it, that the other thing, you can sit here in the present day and you can look in the past, and it's easy to be judgmental. Oh, why didn't they do it like this? Why, they should have known this. Because we have all the expertise and knowledge of today from 25 years in our field. And so we're careful to say this is not meant to be a punitive examination. It's more of an example of like, here's how we should be citing when we move forward, like recognizing these alternative explanations, recognizing that some of these experiments, they did not definitively show this. And so it's not supposed to be punitive. It's supposed to be like forward thinking in terms of recognizing a problem, identifying it, and like, hey, this is a problem. We've got some bias going on, and then moving forward from it. But yeah, it definitely, it's not the citation analysis, there is a degree of subjectivity in it for sure.
Thank you so much. That's really helpful.
Yeah.
Well, thank you so much, Justine. It really amazing paper. It's painful to hear some of about the attacks you're receiving for it. Thank you for addressing that so bravely and honestly, and sharing it with us.
Thank you. And just thank you so much, everybody, for coming and just, I guess, keeping an open mind of what's out there and the stories. And I just really appreciate people just keeping an open mind, asking good questions. Like I said, if there's something that I wasn't able to answer today, please email me. I'm happy to share the paper. I'm happy to continue the discussion over email. And that's as part of like, I feel a little bit, well, not a little bit, but some of the blame falls at our feet. And so I think it's really important in helping get the information out there and sort of correcting and being part of that. Thank you.
Well, thank you so much. And we'll see everybody again in a couple of weeks. And by the way, the recording of this presentation will be available in the next couple of days and will be distributed to all of the participating clubs. Thank you so much, Justine.
Yeah, great.
So could I interrupt real quick before we say goodbye? Can you bring the comments over also? Can we get a copy of the comments? I don't know if I can get it off of my computer.
Yeah, I would love to, sorry, I was gonna say, I would love to have a copy of the comments too. It really helps me like
Yeah.
look at the feedback and think about what people are saying. And I would love a copy too. Yeah.
Sigrid,
Yeah, we can easily save them and we can put them right next to the video recording. We'll put a link there.
Awesome.
And can you do that, Sigrid?
Yep. That's me.
Okay. Well, thank you all for coming. Thank you, Justine. And we'll see you next time.
Good night, everybody.
Thank you for organizing this talk and making it possible for us to hear her.
Yeah, thank you.
Right.
Very much. Have a good night.